This is not a computer-generated model of an alien creature for the next Hollywood blockbuster. What you're seeing is very much earthbound and alive. So this fish is alive, right?
Yes.
Okay.
You can see some of those vascular cells circulating.
Look closely. There's the beating heart. There's the blood circulating. What you're looking at is a 7-day-old zebrafish under the macroscope. And it turns out this freshwater fish from the slow streams and rice paddies of India plays a vital part in the research into human disease.
So he draw again the vessels, and we can go here and have the exact diameter. So then we combine those pictures, and he calculates the area of the vascular field versus the avascular field. And
you can see if the vessels are growing or if it's not
growing.
Yeah,
Exactly.
Zebrafish are important to researchers because in the first few days of life, the progress of development from egg to embryo is the same for both zebrafish and humans. Add to that, zebrafish eggs are clear and grow outside of the mother's body, so scientists can watch the cells divide and form different parts of the body. They can see what happens to the fish if a cell is removed or destroyed. Researchers can also measure the effects of drug therapies. Here at the North Carolina Research Campus, scientists from North Carolina Central University and North Carolina A&T University are using zebrafish to better understand diseases and therapies.
Exactly. For example, with ginger, this is amazing.
Let's talk therapies first, using a pretty common spice, ginger. There's ginger tea, processed ginger, and fresh ginger root. The spice has been used for centuries to calm upset stomachs, but early results show that ginger may treat several diseases. However, not all ginger is the same.
Like for ginger, and there are fresh ginger and dry ginger, but the active compound, the compound in fresh ginger and dry ginger are different.
Oh.
So yeah, so lots of people are not aware of the difference.
The biocompounds found in fresh ginger, called gingerols, appear to improve blood circulation and help treat anemia by speeding up the development of blood vessels and red blood cells.
It increases the number of blood particles. It increases the flow. Like it is good for the future research on human anemic patients.
However, the biocompounds in processed ginger, called shogaols, seem to aid in cancer prevention. Researchers want to understand the chemical profiles of each type of ginger and how the body processes them. That will determine how much ginger a person would need to eat or how it would need to be formulated to gain the most benefit.
So, the different preparation is different. So ginger is not just ginger. It all depends, the preparation of the ginger product.
So now,
and the tiny zebrafish is key to it all. That green mixture is food for zebrafish. Ginger can be added to the water. The zebrafish gradually consume it. Scientists record the effects.
And you can transplant human cancer cell into the fish, and then you can see how the fish interacting with the blood vessel.
Now let's talk disease, and we need to go back to the zebrafish embryo. Researchers can transplant human cancer cells into the fish and look for what allows the cancer to spread or metastasize.
So when we transplant human cancer into the fish, we can see how the cancer cell interacting with our own immune system. So you can label the cancer cell in one color, and then you can see the transgenic immune cells in the fish, and you can start to ask the question of whether the host immune cells actually are hijacked by the cancer cells. And the cancer cell use our own immune system. Because that they were transparent, you can see everything under the microscope in real time. So that is a major advantage.
So when you inject with cancer cells, the blood vessels start to develop. But if you put some drug against it or some chemical compound, that will reduce the blood vessel development. And that is the goal of cancer research. If you can cut down the blood vessels, so the tumor will not develop. They will not get the food, they will not get the oxygen, so then they will not develop. That is the main goal, and if one day we will get there, then that will be the big success, you know.
You've starved the cell, basically.
Exactly, you stop the cells.